Hello viewers, this is Dr. P. Mary Anupama from the Department of Biochemistry, St. Joseph's College for Women. Today let us study the topic nomenclature and classification of enzymes. Contents of the presentation include introduction, then IUBMB nomenclature, that is International Union for Biochemistry and Molecular Biology, and then the 6 main classes of enzymes. Initially, enzymes were named after the substrate on which they act upon. They used to add "ase" as the suffix. For example, you have urea. Urea is acted upon by urease, giving rise to ammonia and carbon dioxide. While we have another example, arginase. Arginase is given to the enzyme which acts upon arginine, giving rise to ornithine and urea. We have one more example, phosphatase. Here phosphatase was the name given to the enzyme which cleaves off phosphate from its substrate. And we also have another example, lactase. Lactase is the name given to the enzyme which acts upon the disaccharide lactose, giving rise to glucose and galactose. Some enzymes, they are named after the substrate and the nature of the reaction that they catalyze. Say for example, we have malate dehydrogenase. Malate dehydrogenase, its substrate is malate and it causes dehydrogenation of malate, resulting in oxaloacetate, and NAD+ is involved in this reaction. So like this, many enzymes, they were named after the substrate and after their after the reaction which they catalyze, as well as after the product on which they act upon. With time, many enzymes were isolated, they were purified, and they are available in their purest form, and a need has arisen wherein they had to give this nomenclature, a systematic nomenclature enabling identification of each of these enzymes. Then came the systematic nomenclature of enzyme, and we have International Union of Biochemistry and Molecular Biology who has proposed this nomenclature of enzymes, where enzymes are divided into 6 main classes. The 6 main classes of enzymes include oxido-reductases, transferases, hydrolases, lyases, isomerases, and ligases. Enzymes are named by giving a 4 numerical digit enzyme commission number. Of these 4 digits, the first digit indicates the type of reaction that they catalyze. The second digit indicates the group that is involved while the enzyme is acting, and then the third digit indicates any additional group that is involved, and the fourth digit, it indicates the specific biomolecule which is involved in the enzymatic reaction. The exception is isoenzymes which have the same enzyme commission code. And here the example is NAD+ oxido-reductase, whose enzyme commission number is 1.1.1.27, and the trivial name is lactate dehydrogenase. We have 5 forms of lactate dehydrogenase which catalyze the conversion of pyruvate to lactate, while the enzyme commission number remains the same. The international classification of enzymes. We have 6 main classes of enzymes, starting with the first one, which is oxido-reductases. They are involved in oxidation reduction reactions. Transferases, they are involved in the transfer of atoms or groups between 2 molecules. We have hydrolases, which are involved in hydrolysis reactions. Lyases, they are involved in the removal of groups from the substrates. Isomerases, they are involved in isomerization reactions, and ligases, they are involved in joining of 2 molecules. Let us now study the main class, first main class, oxido-reductases. These are involved in oxidation and reduction reactions. We have subclasses also. 1.1 indicating the main class 1, that is oxido-reductase, and the next 1 digit is subclass. These act upon the alcohol groups 1.2, these act on aldehyde or ketones. Then 1.3, these act upon the CH bonds. 1.4, they act on primary amines, while 1.5 group, they act upon secondary amines, and 1.6 groups, they are acting on NADH or NADPH. Let us see one best example for oxido-reductases. The enzyme NAD plus oxido-reductase, whose enzyme commission number is 1.1.1.27 and the trivial name is lactate dehydrogenase. It is involved in the conversion of pyruvate to lactate. The first digit 1 is indicating that it is an oxido-reductase enzyme. Second digit, it is indicating that it is involving alcohol groups. Third one digit, it is indicating that NAD plus is involved, while the fourth digit 27, it is indicating that it is the alcohol group of lactate that is involved in the reaction. Let us see one more example. We have isocitrate dehydrogenase, which is involved in the conversion of isocitrate to 2-oxoglutarate, and the enzyme commission number is 1.1.1.41. Here the first digit 1 is indicating that it is an oxido-reductase enzyme. The second one, it is indicating that it is involving alcohol groups, while the third one, it is indicating that NAD plus group is involved, while the fourth digit 41, it is indicating the substrate, that is isocitrate. Now let us study the second main class, transferases. Transferases, these are involved in the transfer of functional groups. Here again we have subclasses and subclasses. Here 2.1, here the first subclass 1, it is indicating that it is involved in the transfer of 1 carbon groups. 2.2, it is indicating that it is involved in the transfer of aldehyde or ketone groups. 2.3, it is indicating that it is involved in the transfer of acyl groups. 2.4, it is indicating that it is involved in the transfer of glycosyl groups. 2.7, it is indicating that it is involved in the transfer of phosphate groups. 2.8, it is indicating that it is involved in the transfer of sulfur containing groups. We also have subclasses here. Enzyme commission number 2.1.1, it is indicating that the enzymes are methyl transferase, that is, they are involved in the transfer of 1 carbon group, and the 1 carbon group is the methyl group which is being transferred. Then we have 2.1.2 indicating that the enzymes are hydroxymethyl transferases. Then we have 2.1.3 indicating that the enzymes are carboxyl transferases. Let us study one example for transferases, that is histone methyl transferase. Here this particular enzyme is involved in the transfer of methyl group onto the lysine residues of the histone proteins, and this is given the enzyme commission number 2.1.1.43. and here the first two, it is indicating that these are transferase enzymes. Then 2.1, the second digit is indicating that they are involved in the transfer of methyl group. The third digit, it is indicating that they are involving S-adenosyl methionine group as the methyl group donor, while the 43, the last digit, is indicating that the methyl group is getting transferred to lysine, and lysine is the substrate. The third class of enzymes are hydrolases, and here hydrolases, they are involved in hydrolysis reaction. Say for example, we have AX, which is the molecule, and in the presence of water, the bond between A and X is cleaved off, resulting in XOH and HA, where water is split up. And these are classified based upon the bond that they hydrolyze. For example, we have 3.1, it is the ester bonds which are hydrolyzed. Then 3.2, glycosidic bonds. 3.4, peptide bonds. 3.5, the other carbon nitrogen bonds, CN bonds. While 3.6, it is indicating that acid anhydrides are hydrolyzed. Now let us see some subclasses of these hydrolases. We have enzyme commission number 3.1.1 indicating that the enzymes are carboxylic ester hydrolases. 3.1.2 indicating that the enzymes are thiol ester hydrolases. 3.1.4 indicating that the enzymes are phosphoric digester hydrolases. And the example here we are seeing is phosphohydrolase whose enzyme commission number is 3.1.3.1, and here the trivial name for this enzyme is alkaline phosphatase, which is involved in the removal of phosphate from the organic molecules, leading to the formation of inorganic phosphate. Now let us study the main class 4, which are nothing but the liases. These are involved in non-hydrolytic removal of groups, and the second digit it indicates the type of group or the type of bond which is broken. So 4.1 it indicates that the carbon-carbon bond is broken. 4.2 it is indicating that the carbon-oxygen bond is broken. 4.3 it is indicating that carbon-nitrogen bond is broken, while 4.4 it is indicating that the bond between sulfur and carbon is broken. We have seen that the second digit is indicating the type of bond which is broken, while the third digit here it is revealing the type of group which is removed. Say for example in the subclass 4.1.1 we see that the second digit one is indicating that it is the carbon-carbon bond which is broken, while the third digit one is indicating that the broken carbon is removed in the form of carbon dioxide. So here it is the removal of carboxyl group by breaking carbon-carbon bond in the form of carbon dioxide. Then we have 4.1.2. Here it is indicating that aldehyde group is removed by breaking carbon-carbon bond, while we have 4.1.3. It is indicating that ketonic acid group is removed by breaking carbon-carbon bond. Let us see one example for liases. Here we have L-histidine carboxylase whose trivial name is histidine carboxylase, that is involved in the decarboxylation of histidine resulting in the formation of histamine. And here the first digit 4 is indicating that they are liases. Then the second digit is indicating that it is acting upon the carbon-carbon bond. The third digit is indicating that the carbon which is removed as carbon dioxide, while the fourth digit it is indicating the molecule which it is acting upon, that is histidine. Now let us see the fifth main class of enzymes, isomerases. Isomerases, these are involved in isomerization reactions. Here the second digit it indicates the type of reaction that the enzymes are involved in, while the third digit it is indicating the type of molecule that is undergoing the isomerization reaction. Here we have subclasses. EC number 5.1, that is indicating that it is racemization or epimerization which is taking place, while 5.2 it indicates cis-trans isomerization. Let us see the example of an isomerase, alanine racemase. The enzyme commission number is 5.1.1.1 and it catalyzes the conversion of L-alanine to D-alanine and vice versa. Now let us see the last of the main classes, ligases. These are involved in bond formation and they require ATP for the new bond formation, while at times the energy source can come from the other NTPs. Now let us see the subclasses. 6.1, it indicates the bond formation between carbon and oxygen. 6.2, it indicates bond formation between carbon and sulfur. 6.3, it indicates bond formation between carbon and nitrogen, while 6.4, it indicates bond formation between carbon and carbon. So let us take the example here where, say for example, it is bond formation between X plus Y, where ATP is consumed and ATP is cleaved into ADP and phosphate, inorganic phosphate. But at times during new bond formation, the ATP can split up into AMP and pyrophosphate, releasing more energy as per the requirement of the reaction. The second digit, it is indicating the type of bond which is synthesized, while the third digit, it describes the bond that is being formed. Say for example, we have 6.3.1. These are the enzymes which are acid ammonia ligases. And the best example here is L-glutamine ammonia ligase, whose enzyme commission number is 6.3.1.2, while the trivial name is glutamate synthetase. Here we can see the synthesis of L-glutamine from L-glutamate, and the ammonia is getting added to L-glutamate, resulting in glutamine synthesis. Let us summarize what we have studied. We have seen the need for classification of enzymes because many enzymes are being purified. Definitely there is a need for systematic classification of enzymes. The 6 main classes with examples we have studied: the oxido-reductases, transferases, hydrolases, lyases, isomerases and ligases. Thank you.